Hello, and welcome to demola. And today's lesson, we are covering 3 in 1. So drug testing, sustainability, and conservation. Gonna be a lot, so we won't waste any time here. Let's get into it and end topic 4. Okay, so we're kicking things off by talking about drug testing. So, as you can see, we have a bunch of different stages. And when we talk about drug testing, we break it up into contemporary, or the new things that we're doing, and then what we did before. So in terms of the contemporary, the new one, you start with preclinical phase. And this is going to be like your test tube studies, your in vitro studies, and then also your animal experiments in vivo. So of course, there are a lot of ethical concerns, especially with animal experiments. Don't typically come up too much in A-level biology. But really in this stage, you're looking at toxicity and efficacy. So can this drug potentially treat something? And also, is the drug going to be toxic to humans? That's what we're mainly doing these stages for. So you can either use human tissues or a closely related animal. Now, we have 3 main phases of clinical trials for drugs, and you need to remember what the phase is and what's happening. So in phase 1, you get a small group of, importantly, healthy volunteers, yeah? And essentially, you're giving them the drug just to see, is it safe, in terms of does it produce any kind of toxicity, and also what are the pharmacodynamics or pharmacokinetics? In other words, how is this drug going to be metabolized and excreted? Next, we're going to go to phase 2. So in phase 2, you get a larger group now, and all of these patients have the target disease. So you're thinking, okay, well, first of all, is this effective? Does it treat the disease? And then also, in this period, you're going to be changing the dose. So when we talk about an optimal dose of a drug, you're thinking, what is the lowest dose that has a therapeutic effect? And that's the one you go for. And otherwise, in this stage, we're going to be noting about side effects. Now in phase 3, this is when it gets a bit bigger. You get your large group of patients with the target disease, and you get a double-blind trial. So in terms of a double-blind, you may be asked to define this. You're saying, okay, both the patient and the doctor or the researcher don't know if the patient is in the treatment group or the placebo group. And just to clarify, placebo is a substance that doesn't contain the active ingredient. For example, a sugar pill. It's always nice to put an example in the exam. And really what you're saying, okay, optimum dose, is that good? Side effects, uses. That's what you're looking for in stage 3. But the main takeaway is double blind and placebo. Removing bias. That's why we use the placebo and the double blinding. Now, we normally get drugs from plants, right? That's the typical place where we find a drug. And a scientist, William Withering, did an experiment with foxglove plants, and he got this substance called digitalis. And what he used digitalis to do was to treat dropsy, or as we call it today, edema. So what we now use this drug for, what that came from digitalis, is as a treatment to kind of reduce fluid around the heart when someone has heart failure. However, the way William Withering did it is he had the person in front of him who was really ill because they had a disease. It causes a lot of swelling, so that's how they would know. And then he would give them the digitalis, and it would just say, okay, do they get better or do they die? Right? And that's literally the reality of it. So maybe if they died, he'd think, okay, we maybe need to increase the dose, it wasn't enough. Or maybe again, if they died, potentially it would be like, oh, that dose was too high, we're going to lower it down. And that's kind of how he went about changing the dose. So you don't really need to know the exact figures for like the success rate. They would probably give you an exam. But the main thing is comparing this to what we do now. So what you're saying, okay, what do both of them do well? Well, both of them determined an effective optimal dose and also monitored the patients in response to treatment, right? But there are a lot more differences. So William Irving didn't do any preclinical testing. He just went straight to the patient group. There was no placebo. All of them got the digitalis suit. He did a lot of trial and error with dosing. So typically in like your phase 2, you'd work on, okay, what's the optimum dose? However, he just kind of went through it, trial and error. No double blind trial, so he knew he was giving everyone the drug. Everyone knew they were getting the drug. And also, he only tested on patients with the disease. So, okay, this next section shouldn't be too long, but we're going to be talking about plant fibers and sustainability. So, we should know plant fibers are quite strong, yeah? Cellulose, microfibrils, lignin, the whole lot. And also know that you should do a core particle about the tensile strength of plant fibers. But what we really want to talk about is we can use plant fibers to make a lot of material, so ropes and fabrics are the go-to. I'd have an exam. However, these can also be made with plastics, yeah? And to make plastics, we need to use fossil fuels. So we need to think, why would we use plant fibers when the fossil fuels or the plastic are more durable, more effective? The plant fibers are more sustainable. So sustainability is the use of materials now to meet the needs of the current generation, whilst ensuring an adequate amount is left or available for future generations. That's our definition, and that's one they can test you on in exams. So you're thinking, okay, plant fibers are more sustainable because plants are renewable. So what do we mean by that? Well, you use a plant, you can go again, you can use the plant fiber again. Yeah. Also just think, okay, plant fibers are biodegradable, less damage to the environment, cheaper to manufacture, better for our corporations. Also just know we can use starch as well. So starch can be made for bioplastics and biofuels as well. So those are just considerations to have in regarding sustainability, but it may. Okay, next topic is conservation. So we're going to do a bit of a dive into this one now. So we need to think extinction, bad. If we have less species or less organisms, we have lower biodiversity, right? And what we've become good at, or what scientists have become good at, is identifying this species is at risk of extinction or they are endangered. So we have different ways of approaching this because obviously we want to maintain biodiversity. So we have in situ conservation, and this might be like a national park that you might see. Now, what's good about national parks or in situ conservation is you get to keep the animal or the species in its natural habitat. So they can adapt, evolve as normal because they're going to have to be looking for their own prey. You're just kind of monitoring the environment. And also you preserve that ecosystem. So those are all good things. However, if we're doing it in our own, like our natural environment, we're going to be more vulnerable to biotic and abiotic factors. So like natural disasters, viruses, the whole lot. Yeah, predation even in some cases. Although they will try to restrict that in the in situ conservation. Also, your habitat can become fragmented. So say if there's one area that we look over, that's now going to be a bit different to the other areas we're looking over. Another thing we do is ex situ conservation. So ex situ conservation involves the use of zoos. And the main thing in zoos is to have captive breeding programs. So when you have a captive breeding program, the main aim is, okay, we're going to reintroduce the species. So we're going to take them from the wild, we're going to do the breeding so that we can increase the numbers, and then we're going to reintroduce them into the wild. And what that does is it builds up the population size, which is good. However, it can reduce the gene pool. So we need to think, okay, if this can reduce the gene pool, leading to, we have to link it back to earlier in the topic, genetic inbreeding or inbreeding depression, we need to think, how do we restrict this? So normally what they'll do is, I'm not sure if you've seen like sheep, they're normally tagged, or you might see like some coloring on their fur. That's gonna say, okay, these sheep either should or shouldn't be bred together, depending on how closely related they are. So that's one thing that they can do, tag different animals. Also, they normally keep like a book that's gonna be like keeping record of the like family lineage of different organisms, and that's also going to help to make sure that we breed less closely related organisms together. Another thing is also sometimes they import from other zoos or swap, and that's going to really help because then you're introducing genes from one zoo, bring into another, you get more alleles in the gene pool. So I know I haven't written those down, but those are also things that zoos do to kind of maintain genetic diversity. Another good thing about zoos is education. So imagine if you've been in a zoo before, there's going to be a lot of information tablets, or maybe even someone going around that's telling you about these animals. Education is really, really good. And also it allows the zoologists to research. Yeah. So they're going to be able to observe the animal, maybe get some genetic information from the animal, which is good. However, you just need to consider that the animal might not act the same in captivity compared to if it was in its natural habitat. Another thing is seed banks. So seed banks are really, really good. They you get a seed from a plant and you store it, right? So in terms of how it's done, normally the seeds are going to be sterilized and cleaned, and then they're going to be kept in cool and dry environments. So let's just quickly clarify something. You are saying cool environments, not cold, cool. That's really, really important. Yeah. If you say cold, they will mark it wrong in the exam. It's cool. Yeah. And the reason we do this is, okay, if it's cool, you lower the enzyme activity, right? So enzymes have less kinetic energy, yada, yada, yada. You don't get germination of seeds. So they stay as seeds. Also dry, bad for bacterial growth. Bacteria like to have a bit of moisture. So if you dry the seeds as well, it's going to prevent that. And this maintains genetic diversity because you're going to keep those alleles. Yeah, you can take multiple seeds from multiple plants, keep them in the seed bank, and that's really going to help. So, like for example, if there was a natural disaster or a virus that wiped out a bunch of these plants, you could then reintroduce these ones, and that would also be good. And then again, the same things kind of come into play. Whereas if it was only the ones in the seed bank, you'd probably end up with reduced biodiversity as well, just because there's less genes. And given we just mentioned bacteria, we just need to talk about bacterial growth to end this off. So in terms of ideal conditions for bacterial growth, you should know these, yeah? Nutrients, warm temperature, moist, oxygen if they're anaerobic bacteria, and on optimal pH. Those are all things that bacteria like. And in terms of bacterial growth, you have this curve, which you should learn and know, yeah? So bacterial growth is split up into 4 different stages. Lag phase, exponential phase, stationary phase, death phase. And you need to know what's going on in each of them. So I have got the definitions down there, but we're going to just show it here. So in the lag phase, you're having a really small increase in the numbers. Yeah, they're getting ready for the exponential phase where they're just going to grow. Yeah, grow, grow, grow. Population size increases a lot. Then you have the stationary phase. So in this stage, the death of the microorganisms, the rate at which that's happening is equal to the rate at which they're reproducing. So you just kind of have this stationary thing. And that's because, imagine, all of our nutrients are being used up in that exponential phase. Yeah, so there's less nutrients, and then the toxic waste is also starting to build up. Yeah, in the death phase now, the toxic waste is like overload, 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 and the nutrients isn't as much, which is why more organisms are going to die than reproduce, and then you get this decline. Importantly as well, I just need to point out log. Yeah, so just know if you ask your calculation, it will probably be in the log. Obviously, read the graph, but it'll probably be in the log, and you do need to convert that. So you can do an exponential function to reverse the log. Now, in terms of what's going on here, you have your definitions there. So you can pause the video, have a read of those. Okay, it's question time. So we've actually got 4. So your first one, you're looking at plant fibers as oil-based, advantage, disadvantage of plant fibers. Yeah. Then you're doing aseptic technique. What's good? This is a practical that you should have learned. Then we're saying, okay, bacteria grow rapidly in skin wounds. Why? And finally, you got a bit of information there. But then we're just talking about how breeding in zoos maintains genetic diversity. So I'm going to zoom out, pause the Okay, so let's look at the answers. So for the first one, I think literally sustainability is like good enough. I would have probably gone with renewable, I think, or plants being regrown. I would have gone with that. I think the other ones are a bit, I think sustainable, it's a tiny bit of a cop out there. Also, decomposed biodegradable, great. I actually really don't agree with this. I don't know why this is under disadvantage. I wouldn't agree with that. I think less strong is what you really want to go for there if it comes up in the exam. Now, the next one was about the aseptic technique. So these are things that you should have known from doing the practical in school. So we flame disinfect the surfaces. That's good. Keep the lids off for a minimum amount of time. Also good. You're thinking flame hoods are good. Disinfecting the bench, autoclaving plates. Yeah. So just note very quickly, sterilizing normally isn't good enough. So I don't want to say like, okay, wash your hands. Autoclave, things that will flame the equipment, anything like that, that's really going to help you. But it's just sterilizing it by itself won't be enough. Next, we talked about why bacteria grow well in the skin. So you need to think back to what were the good conditions for bacterial growth and then link on that. So at warm temperature, your skin's warm, yeah? So that's why it's good. And we had to think again, enzyme controlled reactions, that's going to be the reason. Also, we're thinking, okay, well, what else did they need? They needed an energy source, so a nutrient source. And I normally would kind of state it. So they're going to get nutrients from the blood, probably. So I would just say that. And I would always name one. So I'd probably say like glucose, for example, so that they can respire. Another thing we said, moisture. So you can say there's appropriate moisture on the skin. Yeah, your skin shouldn't be dry. Yeah, so appropriate availability for water. And then you're also thinking, okay, oxygen was another one, aerobic or anaerobic bacteria, whichever you want to go with, but that's also going to be relevant. Finally, in terms of breeding programs, they said, how are we going to like maintain the biodiversity? Yeah. So you're thinking, okay, we talked about selecting specific animals. We did talk about that. And we want to prevent inbreeding depression. Yeah. So that's why we don't breed closely related. The stud book is what we're going for. So I said we have a book that keeps a record of the like lineage. So that's called a stud book. So I'll actually highlight that so it's a bit more clear to you. But that's what we're using. And also we said about exchanging between zoos as well. I know I didn't write those down, so I've included the questions to make sure you can see it. But yeah, these are okay. I hope that hasn't been too long. So I'm gonna whiz through these so I can get you gone from the video. So what is good to do? Stages of drug testing, very good to remember. Yeah. Contemporary versus William Withering, also good. That's a question that comes up very much in AS biology. We also have sustainability. I think the definition is the most important thing in that. So just know the definition. Again, I think definitions are so high yield when it comes to A levels. Importance of conservation, also good. And more and so with linking on to the thing about preventing inbreeding depression or making sure we maintain the size of the gene pool. And also we want to say, okay, bacterial growth curve, it's really good to just kind of know how that curve looks so that you can identify it or draw it should the case be, and remembering those different stages as well. But it's a bit of a less common question, I will admit. Now, what's bad to do? Stages of drug testing, I think, can be mixed up, especially the more you go on in the course and you think, okay, this wasn't as serious. I will admit that's a subject that a lot of people kind of forget about, but it comes up not regularly, but often enough to a point where I think you should remember it. Plant fiber cellular structure, that just links onto what we said about plant fibers and sustainability. They can easily link the questions there. Captive breeding as well with genetic bottlenecks. Again, very easy to link those two together. So make sure you can do it. And then also, this is a big one. Remember, it's a log function on the bacterial growth curve. It will almost always be a log function. So you need to know how to convert. So for example, if it's log base 10, let's just say it's log base 10 of like 4.8. To reverse that, you need to do 10 to the power 4.8. Just remember that. Yeah. Okay. That's all for me. So thank you so much for listening. I hope it was helpful. If it was, please like the video and also subscribe to the channel. Any questions at all that you have, put them in the comments below. I'll do my best to answer in a timely fashion. We've also got our Instagram and our TikTok, both at demola. Links will be in the description below. But that's all for me for now. So thank you so much for listening. I'll catch you.